The overall goal of this procedure is to establish and optimize metabolite and RNA sequencing data from tobacco tree leaf material for systems biology-oriented estimations. This is accomplished by first collecting the biological material of a developmental leaf series. The second step is to perform extraction of the material using the appropriate solvents dependent on the subsequent analysis. Next, metabolites and hydrocarbons are measured using gas chromatography mass spectrometry, or GCMS, and the transcripts are analyzed using RNA sequencing data acquisition. Ultimately, the combination of metabolite profiling and transcriptional profiling via RNA sequencing is used to show how tobacco tree leaves develop long-chain hydrocarbons and other important compounds.
This method can be used to identify and quantify important, useful metabolites made in the leaves of the tobacco tree.
Though this method can provide insight into the tobacco tree metabolome, it can also be used for other systems such as tobacco or related species.
Generally, individuals new to that method will struggle because tobacco tree leaves are tough to extract good RNA from.
In this multi-biopro video, you will see members from the Fernie group extract and profile metabolites using GCMS. You will also see a member from the Fraser group extract hydrocarbons, and also one member from my own group who will extract RNA for RNA sequencing.
Begin by growing Nicotiana glauca plants in 30 cm diameter pots containing M2